for joining us. As you know, this is the Frontiers in Child Health Research Seminar, which we hold on the second and fourth Mondays from September through June. And our series includes speakers with work focused on child health, both from UCSF as well as guest speakers. Please let me know via email if you're interested in speaking or would like to suggest a speaker. This is our last seminar of 2023. We are not going to meet on Christmas. I'm still working on the 2024 schedule, but we will have a speaker on January 8th. And then I wanted to alert everyone that we will have an in-person seminar on January 22nd for the Dr. Walter Miller Lectureship hosted by the Division of Endocrinology. The speaker will be Dr. Richard Auschus, I might have pronounced that incorrectly, from the University of Michigan, and his talk will be titled New Steroids and New Treatments for Congenital Adrenal Hyperplasia. He'll be speaking in the Oberndorf Auditorium from 12 to 1, and there'll be a reception to follow. Otherwise, we're still virtual and using Zoom, and we ask that you stay muted unless you have a question, and we'll also open it up at the end to questions. Today, we're welcoming Dr. Emin Maltepe, who's a professor of pediatrics, biomedical sciences, and developmental and stem cell biology here at UCSF. He's a member of the Division of Neonatology. Emin is also the Director of Product Development for the Initiative for Pediatric Drug and Device Development. His lab focuses on the interface of genetic, epigenetic, and environmental factors during stem cell fate determination, particularly with respect to mitochondrial oxygen sensing pathways and cell fate in the placenta. Disruption of these pathways and other placental developmental functions results in defects in embryonic vascular, cardiac, and hematopoietic development, while other perturbations lay the groundwork for pregnancy complications such as preeclampsia. His research is funded by the NIH, the Department of Defense, and the Bill and Melinda Gates Foundation. The title of Emin's talk today is Developing Oxygen-Targeted Therapies for the NICU and Beyond. Welcome, Emin.
Thanks so much, Roberta. I really appreciate that. Let me get my slides going here. And are you guys seeing my slides?
Yeah, that looks good.
Great. Thanks so much. So I'm just going to jump right in. Thank you for that wonderful introduction, and thanks everybody for joining today. I'm going to start with a slide that, if you don't remember anything about what I tell you today, is really the take-home message ultimately that I would love for you guys to remember, is that really drug development in pediatrics is truly an underrepresented aspect of medicine. If you sort of think about all of the FDA-approved drugs that are out there currently in practice and in use for adults, there's nearly 20,000 of them. There's greater than 19,000, and this is straight from the FDA website. And only about 1,000 have been labeled for pediatric indications, which, you know, at face value is not horrible, but clearly that's less than 5% of the total drugs that have been developed globally. Now, when we get down into my world, into the NICU, the number of drugs that are actually indicated or approved for neonatal disease processes gets down into the 20 to 50 range, which really represents less than 0.25% of all the FDA-approved drugs that are out there. And clearly, we in neonatology are at a significant disadvantage. And if you think about globally, the pediatric market overall, individuals under age 20 represent the largest segment of the world's population. And from an investment standpoint, less than 10% of capital is invested in healthcare innovation targeting pediatrics. And therefore, I think developing drugs for pediatric indications and coming up with mechanisms that will allow that drug development process truly is a key equity issue for all of us in the field of pediatrics to work toward advancing. Now, why is that? Why is that the case? And obviously, many of us know many of the challenges of the drug development process within pediatrics. Namely, one of the major ones, obviously, is patient access is highly limited. There are multiple design restrictions placed on clinical trials within pediatrics in general. A couple of areas that I'm really going to highlight today that I think are critical gaps in the pediatric drug development landscape involve lack of or limited preclinical disease modeling capabilities, and highly importantly, a lack of sophisticated clinical pharmacology efforts devoted to supporting such efforts across the country. Partially, the reason for this is that there really are few training grounds that are actually developing pediatric drug development, developing folks with drug development expertise in pediatrics. And as a result of this, any clinical trial that actually makes it out the door and gets implemented has a low productivity rate. And typically, there's something on the order of 50% of the pediatric clinical trials fail. And this is not with new drugs. This is typically repurposing of old drugs that were shown to be effective in adults. And finally, there's really no one-stop shop that can kind of support pediatric drug development, that can bring all of the relevant expertise and disease modeling capabilities under one roof to enable the drug development process. And this is something about 7 years ago or so that we decided to try to move the needle on a little bit, working closely with the School of Pharmacy here at UCSF and led by Janelle Boyle, who's a professor in the School of Pharmacy here, along with Jeff Feynman, who all of you guys know and has been very active for decades now with pediatric preclinical disease modeling. And we've instituted a formalized agreement in the form of an MOU to create an initiative for pediatric drug and device development between UCSF's Department of Pediatrics in particular, and the University of Maryland Center for Translational Medicine, which is led by Joga Gaburu, who spent about 15 years at the FDA as the head of pharmacometrics. And when he left around in 2012 to join University of Maryland, he founded the Center for Translational Medicine as a way to help primarily companies, but also nonprofit ventures develop drugs for all indications. But he has always had a significant passion for developing drugs and advancing the pediatric drug development landscape. And together with Vijay Vaturi, another senior scientist at the Center for Translational Medicine, actually co-founded one of the leading pharmacometric modeling and clinical trial design software suites called Puma's AI. And we have been working together over the past 5 to 7 years to really try to develop some drugs specifically for pediatric and neonatal indications. And the way we're doing this is really taking this one-stop shop model type of approach where any nonprofit or for-profit biotech or pharmaceutical company can actually come to us at IPD3, and we can enable their drug development process as far early as inception all the way to regulatory submission, given the extensive FDA expertise of the founding members of IPD3. What I'm going to concentrate much more on today is the work that we've been doing with respect to preclinical drug development and how we've been leveraging the resources that we've marshaled to bring together under one roof, under the auspices of IPD3, to drive some of this forward. Now, one of the key elements that I had mentioned earlier in terms of a lack with respect to drug development in this space is the lack of preclinical disease modeling. And that's really an area that UCSF has excelled in for over 50 years. And I have a picture here of Dr. Abe Rudolph, who passed away last year, but he really was the father of preclinical disease modeling for fetal and neonatal cardiovascular physiology. Pretty much everything that we know about cardiovascular physiology in utero and postnatally, came through, at least in part, the work that Abe did here in the Sheep Lab up on Parnassus. And as a result of that infrastructure that Abe and then his colleagues at the time, as well as people like Jeff Feynman, have been continuing to work with, I've been working very closely with Jeff and trying to utilize his unbelievable expertise in the area of large animal disease modeling, specifically in sheep, to enable us to be able to do some sophisticated both clinical pharmacology work as well as efficacy and disease modeling work to see if we can actually get some drugs out there into our patient population. And what I really want to do today is give 3 case studies of the type of work that we've been doing as part of this collective. And the 3 areas that I'm going to concentrate on are an example of a sort of a more classic rational drug repurposing effort. Second, I'm going to talk about what I think is a really novel shotgun drug repurposing effort that we've been doing with the Gates Foundation. And then finally, I'm going to end with an example of some new drug development efforts that we've been doing in partnership with a local biotech. to help them develop drugs specifically for our pediatric indications as opposed to adult indications. So starting with our rational drug repurposing, and I'm going to start with this disclaimer that pretty much I see the whole world through an oxygen-colored lens. A work that I did as a graduate student at the University of Chicago really led me to pursue neonatology as a career. And the work that we did at the time involved creating mouse knockout models of a family of transcription factors that had just been described by primarily Greg Semenza's laboratory at Johns Hopkins, known as the hypoxia-inducible factor family of transcription factors. And these transcription factors were responsible ultimately for driving erythropoietin production in response to diminishing oxygen tensions at the cellular level, as well as at the organismal level. And when we did the knockout of HIF, we saw something really surprising, and we saw that HIF, the absence of a transcriptional response to hypoxia, ended up being embryonic lethal, meaning you couldn't generate mature embryos beyond, it turned out, mid-gestation in this case. It was day 10.5 out of a 20-day gestational period in the mouse. And the reason for this was the blood vessels didn't form properly. And these blood vessels didn't form properly, not because they couldn't produce key angiogenic molecules like VEGF. They just didn't know where to produce them. And the blood vessels initially formed, vasculogenesis was actually present, but if you see in the knockout embryos, either in the yolk sac or in the embryo proper, there is a dramatic reduction of a mature vascular network, both in the embryo, and we later showed as well as in the placenta, that resulted in embryonic demise 100% of the time due to this impaired cardiovascular development profile. And at that time, what that really led us to propose was this idea that oxygen wasn't just a substrate for metabolism, but that it was actually a morphogen. It was actually directing basic biological processes across gradients of hypoxia. And it was those gradients of hypoxia at this tissue and cellular level that were directing blood vessels where to go and how to develop. And in the absence of that positional sensing ability, blood vessels simply did not form, and the embryos died. And over the past 25 years, a whole host of laboratories have contributed tremendously to our understanding of oxygen's role during development. And we now have really come to appreciate this notion that intrauterine existence in mammals occurs in a hypoxic environment, and that this hypoxia occurs at the level of embryo implantation and affects embryonic stem cell proliferation and differentiation decisions. It affects trophoblast stem cell and placental proliferation, as well as differentiation decisions, which I spent many years studying after I finished my initial postdoc as a fellow here in Susan Fisher's lab. And we now know that as a result of this in utero hypoxic exposure, organ development is affected essentially in every organ. And that's because every organ in utero sits at a relative hypoxia. It actually turns out that the most oxygenated organ in utero, because of blood flow patterns coming from the umbilical vein, first going to the liver. The liver turns out to be the most oxygenated organ developmentally in utero, with the brain actually being one of the least oxygenated organs in utero. And we know in each one of these organs, HIF-dependent gene expression plays key patterning roles, regulating both angiogenesis, but also multiple other aspects of organ development. Now, this was really the research project that led to my interest in neonatology as a field. Because if that is actually true, that we all exist in utero in a hypoxic environment, and that there's a complex transcriptional response to hypoxia that's operating in every organ that turns out to be critical for basic patterning, angiogenesis, and developmental pathways, being born prematurely and being exposed to a high oxygen environment as a result of either invasive mechanical ventilation or just simply using your lungs to do gas exchange, which is much more efficient than the placenta, will lead to a premature increase in oxygen delivery to these preterm infants, ultimately inhibiting HIF activity, in a similar fashion to doing a HIF knockout. And this may contribute to multiple of the acronym-based disease processes that we are routinely faced with in the NICU, such as retinopathy of prematurity, bronchopulmonary dysplasia, periventricular leukomalacia, and NEC. And currently, there's good evidence to support a role for aberrant HIF signaling and hypoxia biology in all of these areas. Probably the most well-developed disease process that affects neonates is to involve HIF-dependent gene expression. We now know is retinopathy of prematurity, or ROP. And in the case of retinopathy of prematurity, this exact sequence of events where the preterm infant is developing happily in its hypoxic, warm, wet womb inside the uterus is artificially or prematurely interrupted, resulting in premature birth, and subsequent hyperoxia exposure results in downregulation of the HIF protein and resultant downregulation of multiple HIF target genes, including VEGF, or vascular endothelial growth factor, erythropoietin, IGF-1, as well as some important omega-3 polyunsaturated fatty acids, which it turns out the HIFs also regulate. As a result of this, vessel growth stops, and this is the period of retinopathy that is called phase 1 ROP. And this goes on for several weeks until about 30 to 32 weeks gestational age, where as a result of increasing retinal growth in the absence of proper vascularization, as well as increasing tissue metabolism within the retina itself due to increased activity, results in a reactivation of a tissue hypoxia and a reactivation of HIF-dependent gene expression that then triggers a reactivation of these downstream target genes. However, the revascularization that occurs at this point is not coordinated with retinal growth, and the retinal neovascularization actually ends up looking more like a wound healing type of response and causes a discoordinated revascularization phenomenon, if extremely extensive, can result in scar formation within the retina and ultimately detachment of the retina and ultimately blindness. And this is all called phase 2 ROP. It's the more severe form of ROP. And the only treatment we actually have for this at this time is direct ocular injections of a monoclonal antibody, Avastin, that inhibits VEGF function. We also know that if we control oxygen exposure early in preterm infants' lives, we can prevent the suppression of VEGF and erythropoietin. We no longer do that, however, because a permissive hypoxia in the NICU, for unknown reasons, still leads to some increase of in-hospital death prior to discharge, primarily in small for gestational age infants. And so because of our more liberal oxygen exposure policies, our rates of ROP have gone up. And there are now opportunities, I believe, to actually target HIF signaling therapeutically using inhibitors of HIF-dependent signaling pathways. And the one that I'm going to concentrate most on this morning is one called MitoQ. There are also HIF agonists that are able to turn on HIF that are currently in phase 3 trials globally, as well as in the United States, that are made by Fibrogen, which is literally right across the street from our NICU at Mission Bay, as well as some other compounds. These are less likely to translate clinically in the near term. And MitoQ, on the other hand, is very likely to be able to translate it to a NICU application in the near term. And I'm going to focus on the biology of that for the remainder of this portion of the talk. So what is MitoQ? MitoQ is a mitochondria-targeted antioxidant. This is its molecular structure. The business end of MitoQ is actually ubiquinone, which is a natural superoxide scavenger. It is connected via a linker molecule to another biochemical entity called triphenylphosphonium. The high positive charge of the triphenylphosphonium takes this entire compound MitoQ, because of the high positive charge, it localizes and concentrates very effectively in organelles within the cell that have high negative charge, like mitochondria. So instead of being a global antioxidant like vitamin C or vitamin E, for example, this antioxidant specifically accumulates within mitochondria and works to block hypoxic HIF induction by blocking reactive oxygen species production within mitochondria that are the key signaling component, allowing a cell to sense low oxygen and drive HIF accumulation. HIF, when it goes into the nucleus, it heterodimerizes with its obligate heterodimer called HIF-1 beta. And together, we now know that this transcription factor complex can activate thousands of downstream target genes. And some of the diseases that are impacted by inappropriate HIF activation are pulmonary hypertension, cancer metabolism, various other vascular and metabolic disorders. And MitoQ has actually a very favorable clinical pharmacology, pharmacokinetic profile, where a single oral dose of MitoQ taken daily has a very predictable and favorable pharmacokinetic profile that enables single oral dosing regimens in adults as well as, I believe, children. And in order to test MitoQ's ability to be translated in either preterm infants or term infants with aberrant HIF-dependent signaling driven by various disease processes, I've really been collaborating very closely with Jeff Feynman as well as other folks in critical care, including Sanjeev Datar, as well as Jason Baim, using a model that Jeff, along with Dr. Reddy, our CT surgeon here, developed nearly 30 years ago, about 27 years ago, where they placed fetally in utero, in lambs, a shunt, a graft between the pulmonary artery and the aorta that essentially became a patent ductus arteriosus that never closed, so that in utero development was unaffected and in utero vascular blood flows within the pulmonary vasculature were unaffected. However, postnatally, this resulted in a significant left to right shunt, increasing pulmonary blood flow about twofold on average in newborn lambs, which then was able to generate a lot of the pathophysiology, the pulmonary pathophysiology that we see frequently in babies with patent ductus with a patent ductus arteriosus, or with children with congenital heart defects that have significant left to right shunts, such as large VSDs. You can see on this X-ray from a paper of Sanjeev's that a 6-week-old lambs with this open shunt have significant pulmonary edema and very large hearts consistent with what's seen in this disease state, both in term infants with congenital heart defects as well as preterm infants with patent ductus arteriosus. And it turns out, I'm just going to show key data, but within the pulmonary vasculature, a key driver of pulmonary vascular dysfunction that arises as a result of the biomechanical forces associated with this increased pulmonary blood flow resides at the level of mitochondria within the endothelial cells that line the pulmonary arteries, or pulmonary artery endothelial cells. Sophisticated microscopic imaging of these mitochondria done in collaboration with Steve Black's group in Florida have now shown that the shunted lambs, the endothelial cells derived from them, have disrupted mitochondrial architecture. You can see they're broken up or they're fizzed as opposed to being fused in their control, age match control lamb pairs. We can see in this setting, these mitochondria have altered membrane potential. Their ability to generate energy is reduced. They generate increased reactive oxygen species, and I'm not showing you that data. Importantly, they prematurely activate, they overactivate HIF as a result of this increased ROS coming from these endothelial cell mitochondria, simulating a hypoxic disease state. And we know that adding MitoQ to these cells can blunt or almost abrogate HIF accumulation in a very effective way. And what we did in these animals to test whether or not again, these are newborn lambs with a disease state that's developed initially in utero and then manifesting postnatally, that recapitulates many of the underlying pathophysiology driving some of the pulmonary vascular disease outcomes that we see in both the ICU and the NICU. Jason Baim, as well as Sanjeev and the rest of the folks in the sheep lab, have been able to show now very effectively, and I'm just going to focus on this last section, that pulmonary vascular resistance in these lambs is dramatically, or the response of the pulmonary vasculature to acetylcholine, which liberates nitric oxide from endothelial cells, is restored in these lambs that have seen MitoQ on a daily basis. So this was really encouraging data and really a tour de force of preclinical disease modeling. Another force of preclinical disease modeling has really been spearheaded by Sanjeev Datar, who's been studying the lymphatic biology of these animals. And what he has done with collaborators at the UC Davis Veterinary School has done incredibly elegant CT lymphangiograms to show that the lymphatic system is aberrantly developed in shunted lambs. And he can actually measure flow in this lymphatic system using a sophisticated lymphangiogram washout kinetics, showing that this engorgement of the lymphatics in shunted lambs is due to slow transit of lymph through them. And then when we actually treat these animals with MitoQ, we see a normalization of lymphatic architecture as well as lymphatic flow. And yet another real tour de force of preclinical disease modeling, working in collaboration with Joao Soares, a faculty member in the vet school at UC Davis. They've been able to do sophisticated pulmonary function testing on these lambs, showing that lung compliance is reduced in shunt animals, and that is normalized with just, again, a single daily dosing of oral MitoQ. Now, all of that suggests to us that we've actually got a safe and effective dose of MitoQ that can be utilized, that has been utilized in newborn lambs that are approximately the same size and have the same basal metabolic rates of human newborn infants. And we're beginning to put together a proposal for a clinical trial where we would actually give MitoQ to preterm infants in a multi-site randomized placebo-controlled manner to infants born before 30 weeks gestational age. And depending on the birth date and the feeding status of the infant, we propose to begin treating daily with MitoQ at 28 weeks and continue treating with MitoQ until discharge, looking for incidence of severe ROP as the endpoint, because remember, MitoQ will inhibit HIF, and we believe that HIF activation is responsible for severe ROP progression. And as a secondary outcome, we're very interested in, obviously, its effects on the respiratory needs of preterm infants because of the likely presence of PDA in many of these and the downstream sequelae of PDA in the NICU. So that was an example of how we kind of take classical drug repurposing approaches based on a basic understanding of biological operating processes and design therapeutics in a repurposed manner, specifically for neonatal and pediatric indications. What I want to dive into now, however, is an approach that we've been taking over the past 4 years or so, which is really what we're calling more of a shotgun drug repurposing effort. And this is really attempting to come up with novel therapeutics for birth asphyxia or hypoxic ischemic encephalopathy, which can cause severe brain damage and long-term neurodevelopmental sequelae, including cerebral palsy in kids. And the real challenge with cerebral palsy is that the vast majority of the disease burden for cerebral palsy exists in low and middle income countries. In fact, greater than 95% of patients that have cerebral palsy today reside in sub-Saharan Africa or South Asia. And it turns out the one therapy that we actually can offer newborn infants with birth asphyxia or hypoxic ischemic encephalopathy doesn't work in low and middle income country settings. And it turns out it's not because they can't deliver it correctly, it's because the disease pathogenesis looks different. And this, in my mind, is a landmark study that was led by Sudan Tayal at Imperial College London a couple of years ago. It came out in Lancet Global Health with a large international trial in India, Sri Lanka, and Bangladesh, looking at hypothermia versus control, which showed no improvement in death or moderate or severe disability, which was the primary outcome. However, there was an increase in incidence of death prior to 18 months in the hypothermia group, suggesting that therapeutic hypothermia was not only not helpful, but potentially harmful in this patient population. And therefore, therapeutic hypothermia is no longer considered the standard of care globally. In that setting, given that there's absolutely no therapies that can be afforded and given to children born with HIE in the global setting, we at IPD3 were contracted by the Bill and Melinda Gates Foundation via a research proposal initiated by Farhad Imam originally, who himself was a neonatologist and recruited to the foundation to lead these efforts, and more recently has been taken over by Cicely Fadel, who is a faculty member at Boston Children's, and supported by Maureen Halligan. Now, this is an international research collaborative, including Anka Pasca from Stanford, Hemin Sabir's lab from University of Bonn, and Nikki Robertson from Imperial College London, that was looking to marshal both rodent models as well as large animal disease models in parallel with human iPS-derived organoid models to screen compounds that had potential to be translated in low and middle income settings to prevent neurological severe outcomes in the setting of HIE. And this was really kind of the overarching idea that Farhad and I put together about 4 years ago, I guess closer to 5 years ago. The idea was to look through the entire body of literature of what turns out greater than 1,000 potential compounds that have been shown to have any neuroprotective efficacy in any setting, any disease model, and come up with a short list that we would be able to put through a high throughput rodent brain injury model, as well as in parallel testing them in these iPS cell-derived human organoids, and then test them in much more sophisticated large animal models, including our model in sheep and Nikki Robertson's model in lambs, and then ultimately see if we can help guide translation of clinical trials in the low and middle income country setting by providing therapeutic potential targets and dosing guidelines for them. And this is exactly what we have done. And out of the initial 1,000 compounds that we looked through, and I'll spare you the details of how we actually came up with a short list, but we came up with a short list of 35 compounds, all repurposed drugs specifically for this effort. And those 35, we were actually able to test 25 of them in Hemin Sabir's rodent model. And this was the model. It's a very tried and true model. It's known as the Rice-Vanucci model of acute ischemic brain injury in these pups that are as a combination of artery ligation and a global hypoxia that generates a pretty reproducible, although highly variable pattern of brain injury. And then the idea was to screen side by side in a high throughput manner all 25 of these compounds to see the level of tissue loss induced as a result of this HIE protocol, and see whether or not any of the compounds were actually neuroprotective in this setting. Again, doing them all head to head in the same hands, in the same animal model, in the same lab, to minimize the obvious challenges of trying to compare outputs across different laboratories. And just to kind of give you an idea, one of the huge challenges is to actually do the clinical pharmacology well, so you could come up with dosing regimens that make sense from a human translational standpoint, and then do sophisticated allometric scaling and pharmacometric modeling approaches to come up with appropriate treatment dosing regimens in this rodent model across orders of magnitude difference in body weight as well as basal metabolic rate. And this work was really championed by a recent graduate of the Neonatology Fellowship Program, Yasmin White, who was actually trained with the School of Pharmacy T32 for clinical pharmacology, and has actually been the first fellow in neonatology to do so. But she really did an unbelievable job coming up with the dosing regimen that then Hemin used to test in his rodent model, these drugs side by side, one against another. And here's the result. And this one figure is really an experimental tour de force, where again, 5 drugs simultaneously tested head to head in a rigorous fashion to identify compounds that had a high likelihood of translation in a low and middle income country setting, and figure out which ones were actually the most neuroprotective. And what you can see in red is the control group, the percent brain area loss. And these are actually all of the control pups that Hemin subjected to this hypoxia ischemia protocol, indicating that there's fairly variable, again, this is a variable model, but in general, on average, about a 45% volume loss in the brains that underwent this process. And then you can see the most effective drug in this model at decreasing the brain area loss was caffeine, a drug we use every day in the NICU. The second most effective drug, most interestingly, was a compound called sonic hedgehog agonist, or SAG, that activates the hedgehog signaling pathway that our prior division chief, David Rowich, used to work on, and he's also been a consultant on this project. Importantly, both of these molecules work significantly better than therapeutic hypothermia did in this model. And this right here was the cutoff where none of these drugs actually met the threshold, including drugs like erythropoietin or darbepoietin, which were recently trialed as part of the HEAL trial. So then we wanted to take, the whole point of the pipeline was to take drugs that we've identified in the small animal, high throughput model, and take them now into our low throughput large animal model. And we created a model specifically for this project, again, using sheep, and it was an umbilical cord occlusion. It was a true delivery model. We targeted asystole for 5 minutes, after which point we resuscitated the lambs, and then recovered them and kept them alive for a 6-day course where we assessed their neurodevelopmental outcomes. And then finally, we euthanized the lambs and harvested their brains for immunohistochemical analyses. And the critical component of our model is we can actually perform drug infusion either to the ewe or the mom or the lamb or both, and do sophisticated pharmacokinetic modeling and PK sampling in utero as well as postnatally, along with the entire host of safety and toxicity studies necessary for clinical translational efforts. And a lot of this work was really led by Jana Mike, a key member of the lab and a faculty member in critical care. And as part of this, as I just mentioned, we can deliver drugs both to the mom and to the lamb. We can do intense PK sampling. We harvest the brains and look at markers of inflammation, myelination, and neurological and neuron survival. We do global cytokine as well as inflammatory profiling. And most importantly, we're able to do really physiologically relevant, again, developmentally somewhat relevant neurological monitoring, neurodevelopmental outcomes monitoring of these lambs. And this is just a picture of the lamb NICU that we've developed in collaboration with our colleagues at UC Davis. So this is UC Davis's animal facility. And you can see here we have 2 lambs intubated, being simultaneously ventilated and with invasive monitors, monitoring blood pressures and enabling us to do serial blood sampling and drug delivery. These lambs are able to be recovered and then housed in the barn facility there for a week, at the end of which we harvest the brains, like I mentioned, and we do it again every week. So weekly, we have lambs delivered. On average, we typically resuscitate about 3 lambs a week doing this. And when I mention neurodevelopmentally relevant outcomes, the one benefit of using lambs is that they are neurologically precocious. And as you can see in this video, in this lamb on this side of the screen, This lamb's hind limbs are quite stiff. This is spastic diparesis within the hind limbs, whereas the control twin, which was uninjured, has normal function of that hind limb. Using this large animal model, we then went on to do adaptive preclinical trial design in this large animal model. What that basically means is we start with multiple interventional arms along with the standard of care, which in the LMIC setting is not hypothermia. So we did not subject these lambs to hypothermia. They received standard neonatal recovery. And we were able to test in an iterative fashion a whole host of these drugs. And we were able to discontinue many of these arms because of futility testing at interim analysis, and then add different arms. And what you can see here is the number of lambs that we've actually put through this pipeline to date is really quite large. And some of them, we only went through a few lambs because it became clear that they were not either protective or were actually harmful, and we were able to stop. And others we were able to continue. And again, Yasmin, and I'm just going to highlight the efforts we did for caffeine in particular, because that ended up being the most successful. We were able to do in utero fetal pharmacokinetics, showing very efficient transport of caffeine across the placenta, as well as predicted pharmacokinetics in the lambs postnatally. And we saw that there was really a significant efficacy with caffeine administration compared, for example, with azithromycin here and some of the other agents that we tried in terms of overall neurodevelopmental outcomes. And this was driven in part, we think, by a direct effect of neuroprotection on the brain. Again, Jana has done a yeoman's amount of work on hundreds of brain samples, supported by multiple incredibly talented SRAs within the lab, including currently Ariana. And together, they've been able to show that there is significantly reduced gliosis within the
Did Emin drop out?
It looks like he froze, so maybe he'll be able to reconnect or come back online, hopefully. You're back on him and we can see you.
Are we live again?
Yeah, we got you back.
Great. Okay. So in the last 10 minutes, I'm just going to talk about some efforts that we've been utilizing with a local biotech company to help develop a new drug for pediatric indications. This is a company known as Omniox. It was actually based on technology that came out of UC Berkeley and Michael Marletta's lab. It's a drug it's a protein based drug, or a biologic based on a family of proteins in thermophilic anaerobic bacteria known as HNOx proteins. Now, these proteins have heme as an oxygen binding moiety, but because they are obligate anaerobes, these are not typically used to bind or carry oxygen to tissues. It's typically in these organisms used to bind nitric oxide and sense NO levels because there's no oxygen in these environments. The biochemists at Omniox, however, have engineered this molecule so that it can act as an oxygen delivery molecule, However, with a very peculiar oxygen affinity, and importantly, these molecules, once they bind oxygen, are not reactive to nitric oxide. And this becomes important because hemoglobin-based oxygen carriers were previously developed as oxygen delivery biomolecules, but failed ultimately because of NO reactivity. The ultimate product that we've been testing with them is a pegylated trimerized version of this molecule, which they've been calling OMX. And the interesting aspect of this molecule is that it has an extremely high oxygen affinity. It's a very left shifted compared to hemoglobin, and it actually looks much more similar to myoglobin's oxygen affinity compared again with hemoglobin. And the idea behind the therapeutic application of this molecule is that red blood cells, which have oxygen bound to hemoglobin, when they come into tissues, typically release their oxygen in tissues at not particularly hypoxic PO2s. And when you get into true hypoxia, and what we're meaning here is levels of oxygen at the tissue level that are nearly limiting or actually limiting for mitochondrial electron transport chain function in the single-digit torr range, there's really no oxygen delivery ability of hemoglobin. And that's where OMX kicks in, unloads its oxygen in these severely hypoxic tissues, and actually can reload oxygen in the venous circulation or in the lungs with the rest of hemoglobin. Again, as part of IPD3, we were contracted by Omniox back in the day, and Janelle really led the studies to do the pharmacokinetics of OMX. And the great thing about this molecule, it really has a very nice long half-life of about 2 days, which allows for single daily dosing or every other day dosing, potentially for even some chronic indications. And again, we went back to the sheep lab to test to see whether IV injection of OMX could support cardiac function in the setting of severe hypoxemia. And again, this model, very briefly, is just juvenile lambs, again, large animal pediatric disease model, that are intubated and just made hypoxic as a result of breathing a hypoxic gas mixture where the fractional, the FiO2 goes down from room air, which is 21% to 10%. And then after the animals have stabilized under hypoxic conditions, we injected them with OMX and looked at myocardial function and oxygen delivery, hemodynamics, and other markers of hypoxia. And this was all funded by an SBIR phase 1/2 fast-tracked application from Child Health and Human Development, and led in large part by Jason Baim, as well as with real significant help from people, again, like Sanjeev and Jeff and Rebecca Kameny, who is now down at Stanford. And without going into too much detail, the real, I think, a beauty of the sheep lab is the ability to do classic cardiovascular physiology by placing admittance catheters in both the right ventricle and the left ventricle. Here we're showing the RV. And what you can see, without getting into too much into pressure volume loop analysis, that under hypoxic conditions, this line right here, which reflects the end systolic pressure volume relationship, is shifted down and to the right. And this is called the ESPVR. It's a direct measurement of cardiac contractility, showing that under hypoxic conditions, these animals appropriately over time decrease their contractility because the heart's oxygen demand is so high that it's unable to provide significant contractile effort when oxygen levels become limiting. However, in animals that were given OMX, what we could see that is under hypoxic conditions, the slope of this line, instead of going to the right and down, actually goes up and to the left. So cardiac contractility, myocardial contractility, is not only preserved, but it's accentuated as you would want it to be under a hypoxic disease state where the heart is attempting to increase cardiac output to meet tissue metabolic demand. And I won't go through the effects on the neurohormones, but we were able to specifically look at oxygen levels within the heart using a dye called pimenidazole. And when the dye lights up in these tissues, you know these regions are hypoxic. However, in animals that received OMX, we know the tissue oxygen levels were able to be normalized. So we could deliver oxygen to the heart and maintain its function in a severely hypoxemic state of severe acute respiratory failure. And that work has been published, but more recently, we've been working with the Army. A little bit odd for a neonatologist to be doing, but we've been working with them to help develop novel oxygen carrying agents to potentially act as a bridge to transfusion in a battlefield type of austere environment or a pre-hospital setting in the civilian world. And what we've been testing is whether or not OMX could actually act as an effective oxygen delivery agent and mitigate the severe consequences of hemorrhagic shock. And again, we went back to the sheep lab, and again, this was all funded by the DoD. And what we did is we took lambs. Again, we focus always on juvenile large animal models. We did a controlled hemorrhage model where 50% of the circulating blood volume was withdrawn from these lambs over a 30-minute time frame, after which a single IV bolus dose of OMX or equivalent volume saline was provided. And thereafter, we monitored via invasive hemodynamic monitoring blood pressures, did blood gas analysis in serial laboratories, and ultimately looked at survival in response to the severe lethal hemorrhagic shock model. And for this, we got a lot of help from Mike Smith, a fellow in critical care who has really got some sophisticated statistics and bioinformatics chops. But what we were able to show is that we were able to very effectively, with just that single bolus of OMX, support blood pressure in this animal model of hemorrhage, improve indices of end organ hypoxia. The lactate levels were significantly reduced in animals that received OMX. The base excess was significantly increased. If you remember, base excess is the reverse. And these values were highly significant, and lactate and base excess are routinely used to determine whether or not patients in the setting of hemorrhagic shock require massive blood transfusion. And most importantly, perhaps, we were able to show that there was a really dramatic survival signal in these animals that were exposed to the severe hemorrhagic shock when given just a single IV dose of OMX, as suggesting that this could in fact be a temporizing measure, at least, able to be used in a battlefield setting where it's difficult to actually, from a logistical standpoint, maintain blood products, particularly if transfusion is required over prolonged periods of time. So we're continuing to work with the military to come up with longer time frames and to really define the pharmacokinetics and come up with rational dosing guidelines to be potentially implemented later in a larger scale clinical trials. But what we're very excited about also is we were just funded by an SBIR phase 2B mechanism, again, working collaboratively with Jeff's group as well as Mohan Reddy, to see if we could use OMX in the setting of infants undergoing cardiopulmonary bypass, where the OMX would actually be put in cardioplasia. And this here is a cardioplagia bag containing OMX. Cardioplagia, for those of you who have seen it, is a clear electrolyte solution with no oxygen carrying capacity. And we're testing currently whether or not OMX could act as an oxygen carrier in infants that are being put on bypass for repair of their corrective heart surgeries, and ultimately decrease some of the downstream sequelae seen in the CICU as a result of babies being on bypass. And so stay tuned. We're hoping that if this set of studies is successful, we could potentially, and we're currently doing, working towards IND-enabling studies to go after an FDA labeling indication for OMX specifically for this patient population. And this would be, again, one of these rare instances where we may have been able to successfully develop a drug for a pediatric indication first before rolling it out to the wider adult world. And as you can imagine, bypass is way more common in adult medicine than it is in pediatrics. And I just want to end with this right here. This was a recent commentary article that came out by Keith Yamamoto, Victor Zhao, and Keith Wailu in Science recently, where they were very astutely arguing that we embed equity throughout the innovation cycle. And the quote from that was that equity is not baked into the nation's innovation process. And I wholeheartedly agree that it's not. But truly, what I would like to extend today and hope to leave you guys with is that pediatrics typically is not baked into the nation's innovation process. And we really need children's hospitals as well as academic departments of pediatrics to forge public-private partnerships to lead pediatric innovation efforts to help get therapeutics to our patients. And with that, I'm just going to thank a whole host of people, at least show their names. I've tried to thank everybody who contributed significantly to this work, and the funders that are responsible for paying the bills. So I thank you guys all for your attention. I'm happy to take any questions, and I think I'm on time, but maybe not have left too much time for questions.
Yeah, we had a little technical difficulty, so we still have a few minutes, and certainly if people have a couple questions after, we could stay on. Does anyone want to speak up or?
Could I ask one, Roberta?
Yeah, go ahead. Mine's pretty mundane, so you go.
Emin, that was quite a tour de force. That's amazing.
Thank you.
How you're pushing things. Congratulations. Maybe more from the clinical trenches, but feeds back to your approach. So just, quote, cerebral palsy. To what extent is that a heterogeneous condition? Are there, subtypes, other explanations that
Absolutely
you can feed into your kind of preclinical models to help explore therapies?
Yeah, thanks for raising that. I really didn't have time to go into it. The response to HIE in neonates is highly varied. And really severe HIE only results in sort of the constellation of neurodevelopmental impairments that ultimately is called cerebral palsy, and something on the order of like 30% of the patients that had severe HIE. And our large animal model actually recapitulates those exact percentages of outcomes. And what we're currently doing is actually taking the lambs that were exposed to the same pathologic insult but recover, and compare those to lambs that don't recover and have severe outcomes versus moderate outcomes. And we're doing various approaches like metabolomics, proteomics, as well as spatial transcriptomics to identify what's different. Like, why do these animals, and one of the beauties, I think, but also the challenges of using the lamb as a disease model is they're outbred, but so are our patient population. And so from that respect, it actually is a great model to do disease trialing methodologies and develop really diagnostic approaches for prognostication, which we would love to do. So thanks for that question.
Yeah, thank you.
Jim, you have your hand up?
I do. Emin, thanks. That was a great talk.
Thanks, Jim.
As you know, I'm interested in preclinical studies as well. So I'm going to ask you a friendly question on your take on something.
Sure.
So sheep are very precocial.
Correct.
Rodents are very altricial. Humans don't lie in between. And
yeah.
What's your take on the current environment at FDA for species requirement to get past the valley of death from really good preclinical work like we both have and actually a chance to try something?
Yeah, that whole valley of death question and the FDA's role in both contributing to it and trying to, deal with it is, we could have a whole separate talk specifically on that. Unfortunately, the FDA, there were, well-intentioned congressional mandates that came out with like PREA and BPCA that really helped, with the goal of trying to advance pediatric study plan implementation in the FDA drug development process. And as you very well know, because you're asking the question, most of these companies either find ways around that or they do really rudimentary rodent studies that actually have no clinical translational potential in order to get that 6 months license exclusivity that process enables the drug development companies. I'll tell you, the thing that just happened in terms of the Inflation Reduction Act, which is going to make this valley even greater and deeper, is the pricing restrictions that are coming really suggests and is really making it such that small molecules, what's actually being called the poison pill, small molecule development is really going to slow down. And the pharmaceutical industry, which, you know, really wasn't looking much at the pediatric population, certainly not at neonatology, is not going to knock on our door at all. And this is really where the academic centers really need to take the lead and come up with creative approaches to advance the landscape of therapeutics design. Because the for-profit sector is not coming, and the FDA, unfortunately, is not able to really do anything about that, or unwilling. I won't say unable. They could if they really wanted to.
Do you think we could get any traction under the argument of orphan patients?
For sure.
The rare disease, particularly thinking of HIE.
Well, you know, people have tried and they're trying, and there's one or two companies out there fighting the good fight. We'll see. It's a challenging, therapeutic hypothermia has been wonderful in some respects. It's given us something to offer, but it's really complicated the interpretation of clinical trials data with respect to new drugs coming into the space, as we've seen with EPO. We just don't know if EPO on its own would have been therapeutic in the setting of HIE without hypothermia. In addition to caffeine, as I mentioned, being trialed in South Asia, it looks like they're probably also just going to try EPO on its own in the absence of hypothermia in that patient population to see if it will be effective. I actually think they need to develop therapies in utero before the infant is born, because all the data suggests the injuries happen significantly earlier and it's not a perinatal insult. And this is why we've developed our sheep model, so that we could actually deliver drugs to the mom, look at transport kinetics across the placenta, and look at safety and toxicity in utero as well as postnatally. But jury's out. It's a tough nut to crack.
Great stuff. Thank you so much.
Thank you.
All right, any final questions? I don't see anything in the chat, unless I'm missing something. No chat. All right, well, thanks so much for joining us, Emin, and we will see you guys in January.
Thanks, everybody. Really
Bye,
appreciate it.
everyone. Have a nice holiday.